All right, let's go ahead and get started. So on Wednesday, we weren't able to actually complete the entire lecture. And for the sake of time, we're not going to actually go over those last few slides. But what I wanted to point out to you was in the slides before that, we talked a lot about the cytokines and how the various cytokines could affect the immune response. The last few slides, basically what they're trying to show you is that in addition to affecting the immune response, these various cytokines can also affect other organs. For example, some cytokines can have an effect on brain functions, have an effect on the liver. In addition, these cytokines can also affect different cell types. And one example is that is actually in your slide is showing that some of these cytokines can actually stimulate endothelial cells to produce new blood vessels. So the main point of those slides is that these cytokines not only play a big role in controlling the immune response, but also can affect other important functions of the body. In today's lecture, we're going to talk a little bit about some of the cells involved in the cell-mediated immune response. Now, we've touched on a bunch of these different cells in the previous lectures, but we really didn't talk about how these cells are activated, what their function, and what their exact functions are in the immune response. So we're going to spend a little time talking about the T helper cells and how the T helper cells play a critical role in dictating what type of immune response is elicited. Is it going to be a humoral immune response, or is it going to be a cell-mediated immune response? We'll also talk about a number of the effector cells and how they get activated and what they do, how they elicit their response. So we'll talk a little bit about the macrophage, talk more about the T cytotoxic cells, and we'll also introduce NK cells, and finally we'll discuss a little bit about how the immune system is regulated. So how do we start from a baseline, go up to an immune response, and then end up somewhere in between. Okay? So you've heard a lot about T helper cells and how they play a critical role in determining the immune response. First of all, they can select which effector mechanism is going to take place. So is it going to be a humoral immune response, so an antibody response, or is it going to be a cell-mediated immune response? So they play a role in deciding which one of those arms of the immune response is going to be initiated. They do this in part by inducing the proliferation of the proper cell. So they can induce the proliferation of B cells. They can induce the proliferation of cytotoxic T cells. In addition to inducing the proliferation, they can also enhance the function of these cells. So once the CTL becomes activated, they can produce cytokines that can actually enhance the ability of that CTL to do its job. And shown here is a pretty simplified cartoon. Again, shown in the middle here is a T helper cell. And as you remember, one of the critical steps in activating a T helper cell is the recognition of an antigen in association with class II MHC. So once the T helper cell receives its signals, depending on the type of signal and in the cellular environment in which it's in, it can then decide to initiate a B cell response, or it can decide to initiate a T cytotoxic cell response. In addition, the T helper cells produce a number of different cytokines that can activate granulocytes, macrophage, as well as NK cells. So the point here is that the T helper cells play a central role in deciding which type of immune response is going to be elicited. And if you look at the development of the T helper cells, it turns out that they can turn into at least two different types. And we'll go through their development. All the T helper cells will start as naive T helper cells. And at this stage, they can primarily produce IL-2. Upon receiving some type of stimulation, they become Th0 cells. And at this stage, they can produce various different cytokines, including cytokines that are important for the cell-mediated, as well as producing cytokines that are important for the humoral response. From this stage, they can then differentiate into two different types of cells, Th1 cell and a Th2 cell. The Th1 cell, as we mentioned before, plays a critical role in the cell-mediated immune response. And as you can see here, these cells can produce cytokines such as interferon gamma and IL-2. And as you'll remember, interferon gamma and IL-2 play a very critical role in stimulating the cell-mediated immune response. In addition to becoming a Th1, they can become a Th2 cell. And these cells play a critical role in the development of the humoral immune response. And again, that's characterized by their cytokine production. As we mentioned before, IL-4 and IL-5 play a critical role in the elimination of the helminth infection, the parasitic worm. Both of these cells, after they become activated and they elicit their immune response, both of them then can become memory cells. So the main point here is that T helper cells, there's two main types, T helper 1 and T helper 2. The T helper 1 plays a critical role in the development of the cell-mediated immune response by activating macrophage or cytotoxic T cells. And the Th2 can play a very critical role in the development of the humoral immune response. Now, in addition to when you have the Th1 or Th2 cell, Th1 plays a very important role again in initiating the cell-mediated immune response. Th2 plays a critical role in the humoral immune response. There is also cross-regulation. So if a Th1 cell is activated, in addition to producing cytokines that activate the cell-mediated immune response, they can also produce factors that inhibit the Th2. And the same is true for the Th2 cells. So upon activation, two important cytokines that are produced are IL-4 and IL-5. These activate the humoral immune response. In addition, the Th2 cells can produce another cytokine known as IL-10, which is important in inhibiting the Th1. Now, if you think about it, this makes a lot of sense. If you have a bacterial infection, you want a Th2 response to be the main response. So if you have Th1 cells also being activated, it's kind of a waste of energy. So it's important to also downregulate the alternate response. At the same time, you're upregulating the appropriate immune response. Sure. The last part? Yeah. Well, basically what happens, you have Th1 and Th2 cells. Th1 is important for the cell-mediated, Th2 for the humoral. And in addition to producing cytokines that upregulate the appropriate pathway, so Th2 produce cytokines that are important for the humoral, they also produce cytokines that will inhibit the Th1. And this is important to produce the most effective immune response. Is that clear? Okay. Again, we're going to go through a lot of different cell types and their role in the immune response. We briefly went over the T helper cells. Their role is much more involved than what I just showed. But now we're going to switch gears a little bit, and we're going to talk about the macrophage. It turns out the macrophage can play an important role in a number of different aspects of the immune response. They can play, as we discussed, a role in the natural immune response. So if they see a bacteria, they can gobble it up and clear it. They also play a very important role in the specific immune response. Again, so macrophages are involved in the initial defense, and they're also involved in antigen processing and presentation. That's shown here in the cartoon. So if you have a pathogen such as a bacteria, without any antigen presentation, these cells can nonspecifically engulf these. That will lead to the activation of the macrophage and production of cytokines. Macrophages can also act as antigen-presenting cells. So upon engulfing this bacteria, they can process it and present it with the class 2 MHC, then present this to a T helper cell. The activated macrophage can also play an active role in clearing the bacteria. So upon activation of T helper cells, they can produce various cytokines and lipokines, which then activate this macrophage, which will help it clear. So again, they're involved in the nonspecific immune response by nonspecifically engulfing bacteria. They're involved in a specific immune response two ways. One, They can act as antigen-presenting cells, which leads to the activation of T helper cells. In addition, they can become activated themselves, which will lead to the efficient clearing of these pathogens. Yes?
Macrophages always have to be activated by T cells to release the cytokine?
No. When they're involved in the specific immune response, yes. But also, just by engulfing these in the innate immune response, they can become activated without the help of the T helper cells. The cytokines produced here are different than the ones produced here. The cytokines produced here lend to increased activation of the innate immune response. So here we'll go through some of the detailed functions of the macrophage. Now, the point here is you don't need to remember every one of these functions of the macrophage. The point here is to show you that macrophage, in addition to being involved in the innate as well as the acquired immune response, also can play a role in other functions. So as we discussed, they can be involved in inflammation and fever production. Typically, this is part of the innate immune response, the very initial immune response, by production of various factors such as IL-6, TNF-alpha, IL-1. We've discussed a lot of this. Here, the role in the specific immune response, or the acquired immune response, is they can actually help to dictate which type of response is elicited. They can produce IL-2, which plays a critical role in the activation of the Th1 cells, which then leads to a cell-mediated immune response. They can increase the IL-4 levels, which leads to the activation of Th2 in the humoral immune response. And also they can be involved in the processing and presentation of antigens. Kind of here's a non-related or unrelated to the immune system. The macrophage can be involved in reorganization of tissue. They have a lot of degradative enzymes that can basically digest a lot of the extracellular matrix. They can also produce factors that are involved in, if you can see this, stimulation of angiogenesis. Again, these aren't very important. Exactly what's going on is not important for this lecture or for what I want you to know. But again, they can be involved in other things like damaging tissue. They have antimicrobial action by production of free radicals. And they also can have anti-tumor activity. So the basic summary for this is that macrophage, in addition to being involved in the innate as well as the acquired immune response, can also play an important role in other cellular functions. Okay? You don't need to remember each one of these. So how are the macrophage activated? Turns out that there are two major ways to activate a macrophage. The first one is by interferon gamma production by a T helper cell, plus exposure to a bacterial endotoxin such as LPS. So they see TNF and then they see bacteria. The other way is, again, interferon gamma production, and also and then in combination with seeing TNF-alpha. So again, what we can see from here is that interferon gamma plays a very critical role in the stimulation of macrophage. Shown here is another cartoon basically showing you these two different ways of activation. T helper cell, after seeing an antigen in context with MHC class II, will produce interferon gamma. Interferon gamma in combination with, for example, a bacterial endotoxin such as LPS can lead to the activation of this macrophage. Now, in addition, an activated macrophage or another cell type can produce TNF-alpha, and that in combination with the interferon gamma can lead to an activated macrophage. So you see here primarily these two signals, interferon gamma combined with either a bacterial endotoxin or with TNF-alpha, leads to the activation of the macrophage. Is that clear? Okay. In a number of the lectures, we talked about cytotoxic T cells. We never really talked about how they do what they do. So how do cytotoxic T cells kill target cells? So when the CD8 cell, or the cytotoxic T cell, leaves the thymus, at that point it's able to recognize the MHC class 1 molecule in association with a peptide. But at that point it's not able to actually kill the target. It needs the second signal. So again, to become activated, it requires the second signal. So again, the first signal is the interaction of the T cell receptor with the MHC class 1 molecule and the appropriate peptide. And the second signal typically is exposure to cytokines such as IL-2 or TNF-alpha, or interferon gamma. Sorry. And that's shown basically here in this cartoon. So the free T cell, when it leaves the thymus, is able to recognize MHC class 1 molecule in association with the peptide. That is the first signal. In addition to that, it needs help from a T helper cell. So the T helper cell can recognize a peptide in association with class 2 MHC. This leads to the production of interferon gamma or IL-2. Combination of the recognition of the peptide in association with MHC class 1 and the interferon gamma fully activates this T cell, so now it's able to recognize the target cell expressing the appropriate MHC class 1 molecule in the peptide. This recognition leads to the killing of the target. So again, it requires two signals. This T cell must be able to recognize the MHC class 1 molecule in association with the peptide, and it has to receive help, either two examples here are interferon gamma or IL-2. That leads to a fully activated T cell that is now armed and able to kill the target cell. Go through some of the features of the cytotoxic T cell killing. They're antigen specific. Again, the T cell receptor has to recognize the appropriate antigen with the MHC class 1 molecule. Important feature here is that in most cases requires cell-to-cell contact. That makes sense. In order to recognize the peptide in association with MHC class 1, there needs to be some contact between the cells. Interestingly, each T cell is capable of killing many targets. So if a T cell goes in, finds the target expressing the right peptide, it can kill that target. Once it kills that target, it can go on to kill another target. They're very efficient at killing. The mechanism of killing, there are several different mechanisms. You can have direct cell-to-cell signaling via surface molecules. We'll discuss this. The two of them are interaction between Fas ligand and Fas, and also TNF receptors. There are some ways of indirect signaling via cytokines. We'll discuss these in more detail in the next couple of slides. And one of the very important ways in which they kill is by using a system that employs perforin as well as granzymes. The perforin basically will poke a hole in the cell, leaving access for the granzymes. Granzymes will go in and cleave the nuclear contents, leading to apoptosis. I'm speaking too fast. I hear a lot of clicking. I know you guys type faster than I do. Okay, so in this system here, upon contact with the right target cell, the cytotoxic T cell will release perforin. Perforin will poke the holes in a target cell, after which that will be followed by the granzymes. These granzymes are enzymatic molecules that will go in and cleave DNA and other components of the cells that are important for its survival. And this will lead to the induction of apoptosis. Does everybody remember what apoptosis is? Programmed cell death? Okay. As I mentioned, granzymes, basically serine proteases, they enter the cell, and then they lead to the apoptosis of the cell. It turns out that apoptosis is very important because there's generally thought of two ways you can kill a cell. One is through an insult to the membrane, so the cell explodes. And if that happens, everything in that cell is released into the environment. However, if it is killed by apoptosis, this is more of an order process where the cell basically keeps everything in the capsules. It basically cleaves everything up, and everything remains in capsules. So the content's not actually released into the environment. And this would be important for example, a viral infection. If you just explode the cell and you release a virus, you still have problems with the viral infection. However, if you keep it enclosed in the vesicles, other cells can come along, engulf those vesicles, and clear it from the body. And as always, with a cartoon trying to explain to you, I know I'm better with pictures. Words never work with me. I need pictures. So shown here is the mechanism of T-cytotoxic cell killing. First step, release of the perforins in association with calcium. This pokes holes or makes channels in the target cell. The cytotoxic T cell will then release the granzymes. They enter through these holes, basically chew up the cell, lead to apoptosis. That kills both the cell as well as any viral particles or bacteria within the cell. It's a very efficient way of killing and clearing these pathogens. After the enzyme enters, basically it will lead to the activation of caspases or other enzymes, and it will end up cleaving a lot of the important proteins as well as DNA within that cell, and that leads to a process known as apoptosis. And in addition, it will also kill the viral particles, the same type of mechanism by chewing them up. Does that make sense? Okay. I probably shouldn't have introduced that term, but caspases are enzymes that are involved in the induction of apoptosis. So once they become activated through another ordered process, they lead to the cleavage of a number of important molecules involved in the cellular processes. So they can cleave DNA, they can cleave other enzymes that are important for survival. Yep. Yes. So here, basically these cells they're attached to each other. This is just shown so you can actually see the order process. So if you actually saw a picture of these two cells, they would be right on top of each other. Yeah. So again, killing by these cells using this method requires cell-to-cell contact. Another method of killing is through FAS, or tumor necrosis factor receptors. FAS and tumor necrosis factor receptors are located on the target cell. So they're signaling through these molecules. So it's not actually an insult to the cell. There's no holes poked into the cells. But these FAS, or the TNF receptors, are expressed by the cells. The target cell will come along, having the appropriate ligand, will bind to them, and that will start the signaling process. I'll show you that in another cartoon. Again, the cytotoxic T cells express FAS ligand on their surface. FAS ligand can interact with FAS, which leads to the induction of apoptosis. TNF, which is the ligand for TNF receptor, can actually be released by the cytotoxic T cell. So this is a soluble factor that can actually kill these cells. Shown here is a figure of FAS ligand and TNF receptor. FAS ligand, again, is expressed by the cytotoxic T cell. It can interact with FAS on the target cell. FAS has what is known as a death domain. Activation of FAS leads to activation of the death domain, leads to a series of events that leads to the induction of apoptosis. So again, the difference here between this and the perforin granzyme is that there are no holes actually being poked into the cells. The result typically is the same. You have induction of apoptosis. Again, you have the TNF and TNF receptor. TNF can bind this receptor and activates the induction of apoptosis in a very similar pathway. The ultimate result here, again, is the induction of apoptosis. Important here in this process is the presence of this death domain. There are also other receptors that combine TNF that don't have the death domain, and this will not lead to induction of apoptosis. The importance is shown here. So again, this is FAS. On ligation with FAS ligand, activates a number of different processes, a number of different domains. Shown here, you have FAD, which is FAS-associated death domain. So that's the death domain here. This activates caspase 8, which leads to caspase activation 3, 6, or 7, induction of apoptosis. Now again, the exact molecules or the exact caspases here are not important for us for this lecture. However, what you need to know is that ligation of FAS, the FAS ligand on the cytotoxic T cell, leads to activation of the caspase cascade and induction of apoptosis. Similar for the TNF receptor. Ligation of this leads to activation of its death domain. It's called TRAD, which is TNF receptor associated death domain, and activation of the caspase cascade. Okay? Does that make sense? So two main ways that we discussed so far: perforin granzymes and through what are known as death receptors, FAS or TNF receptor, both of which lead to apoptosis. Yes?
The receptors are all in the cell that's going to be killed.
Yes. Well, FAS and TNF receptor, yes. FAS ligand is expressed by, yes.
FAS ligand is on all cells?
FAS ligand is not on all cells. It's primarily on cells like cytotoxic T cells. NK cells can also express. But FAS or FAS ligand? FAS is expressed by most cells.
FAS.
Yes. Okay.
But how does this FAS ligand?
As FAS ligand? Typically, FAS ligand is expressed by activated lymphocytes, like cytotoxic T cells. So it's expressed on the surface. So when this cell comes into contact, so it recognizes MHC class 1 in association with the right peptide. That will then allow FAS ligand to, that typically will activate this T cell, upregulate FAS ligand, and that will then lead to the killing. So the point here is that the cytotoxic T cell has to be activated, typically, in order for FAS ligand to be upregulated. There may be some small levels expressed, but to have functional levels of FAS ligand, they have to be activated. So however, how we discussed before about activation of the cells, those processes have to take place first. Then you have activation, then upregulation of those different molecules. Yes?
Just to reiterate, FAS and receptor are on this.
Yes.
Okay.
Yes. I've done a good job of confusing you. If you guys have any questions, just really email me because I typically will respond pretty quickly. And again, that way I can go back and check in my books and make sure I give you the right answer. You might be safer that way. Okay, so let's again talk a little more about the steps to make sure you understand the steps involved in the killing by cytotoxic T cells. First part is the T cell must recognize the antigen on the target cell. Okay, that's a critical part. You don't have for most people, you don't have non-discriminate killing. There has to be the association with the MHC class 1, the peptide, and the T cell receptor. After which, this activated or armed cell will deliver its lethal hit, and this can be perforin granzyme, it can be FAS ligand, or it can be TNF alpha. We'll kill the cell. Basically what it says here is that this cell will detach, and then the cell will die. So what happens, this will deliver the lethal hit, it'll take off. So the lethal hit basically starts the process, the cell will then leave, this cell will die, this cell will go on to try and find more targets to kill. Okay? Okay, so let's switch gears a little bit. There's also another cell type that's a very efficient killer cell. It's called natural killer cell. Now, this differs from the cytotoxic T cells in a number of ways. This cell lacks most markers that distinguish it from a T cell and a B cell. Does not express, for example, CD3. Doesn't express B cell markers. Does not have T cell receptor. Does not have to undergo thymic maturation. What's the main, our most important part of thymic maturation? What happens in the thymus? Yes, yeah, so that's where the T cells are developed. That's where they learn to recognize self from non-self. Turns out that the NK cells don't necessarily recognize self from non-self. They have other ways of recognizing their targets. We'll discuss that in a little bit. Important point here is one way of distinguishing them is that they express CD56. An important part here is that's how we tell which cells in our system are actually NK cells. Also, they express CD16. This is important because this is an Fc receptor. Now, if you remember back in your earlier lectures, Fc is expressed by immunoglobulins. So, by having an Fc receptor, NK cells are able to recognize that portion of the antibody. So they'll be able to recognize any cell or any pathogen that's coated with antibodies. Also, an important point here is that if NK cells are exposed to IL-2, they become what are known as lymphokine-activated killer cells, and these cells have a much greater capacity to kill targets. So you can activate NK cells. Once they're activated with IL-2, they become lymphokine-activated killers, and they have a much greater capacity to kill targets. And this has actually been used, I think I mentioned it a couple lectures ago, this has been used therapeutically to try to kill tumors because lac cells, for some reason, are able to recognize a variety of tumors. So they actually have clinical trials, or actually this has been used clinically to try to treat different tumors. Examples are malignant melanoma and renal cell carcinoma. So by using high doses of this, they can activate lac cells, and these lac cells, in many cases, can find and kill these tumors. Now, the mechanisms of killing are very similar to that of a cytotoxic T cell. They can have perforins and granzymes. They do express fast ligand. The big point here is that they're not MHC restricted. And actually, the mechanism of killing is actually completely opposite. So the cytotoxic T cell will recognize MHC class 1 and associate it with a peptide. NK cells typically recognize the absence of MHC class 1. So if a cell, for some reason, stops expressing MHC class 1, NK cells will see that cell and recognize it as having a problem, and then it will kill that cell. Turns out this is important because a lot of times viruses or different tumors can lose expression of MHC class 1. That helps these NK cells recognize these types of cells. And as I discussed briefly earlier, another method of recognizing and killing the target cells is that a lot of times when antibodies are produced, they'll cover the pathogen. NK cells can come along and recognize the Fc portion of the antibody. The Fc portion will bind to CD16. That will then activate the NK cell to kill that target. So two methods. One, lack of MHC class 1. That will stimulate the NK cell. Or that will lead to recognition by the NK cell. And then antibodies, if a pathogen is coated with antibodies, the NK cell can recognize CD16, which binds to its Fc receptor, leads to killing. So those are the two main ways in which an NK cell can recognize pathogens. And as we mentioned before, stimulation with, for example, IL-2 can lead to a separate type of NK cell known as a lymphokine-activated killer, or a LAC cell. And this significantly enhances the ability of these cells to kill. And in particular, it helps them to kill tumors. Okay, in the last part of this lecture, we're going to talk a little bit about how the immune system is regulated. I think we spent a lot of time trying to discuss how you can increase the activation of the immune system. But it's also important that after the infection is cleared, you have to go from here back down to here. If not, I guess the one point is there's very limited space in our bodies for the immune cells. So if every time we had an infection, we produced millions of cells, and those cells just stuck around, after a few infections, there'll be no room left for additional immune responses. In addition, we have a lot of activated cells running around that will then potentially lead to autoimmune disorders. There has to be some type of mechanism for the downregulation of the immune response. So these are kind of very vague definitions of what's going on. And the reason that is because it's very complicated. It really relies on a number of different factors. So I'll just try and give you a couple of examples. So the magnitude of the immune response is typically determined between the extent of lymphocyte activation and tolerance. So we discussed a lot about activation and how they're activated. We just haven't discussed a lot about tolerance. I think Dr. Ghaffar will spend a lecture discussing that. But what we did discuss is that in order for activation, you typically need two signals. And if you receive the first signal without the second signal, in many cases that can lead to tolerance. So the extent to which you have activation versus tolerance can play a major role in the downregulation of the immune response. Again, so lack of co-stimulation can lead to tolerance. So also, whether you have activation or downregulation depends a lot on which cell types are activated. Is it Th1 or Th2? Again, Th2 can downregulate Th1. Th1 can downregulate Th2. Now, in addition, I'm going to briefly talk about another cell type that plays a very important role in the downregulation of the immune response. And these are known as T regulatory cells. The activation of T regulatory cells actually leads to downregulation of the immune response. Important point here is that you can have regulation at every aspect of the immune response. You can have regulation during activation, during the effector phase, as well as recognition. So all three. The first part is recognition. You can have regulation there. During activation, you can have regulation, and during the effector phase. And this is typically depends on the microenvironment. So if you have more stimulatory signals than the negative signals, you'll have an activation response. If you have more negative than activating, you'll get a negative or downregulation. So in the last part of this talk, I want to briefly introduce to you a relatively new cell in the immune system. They're called T regulatory cells. These have, I'd say, in the last 5 or 10 years, maybe. I'm going to ask the two guys back there. They probably know better than I do. I'd say the last 5 to 10 years, they've been recognized and identified. This is a cell population that expresses CD4 and CD25. And CD25 is IL-2 receptor. For the most part, these cells are also generated in the thymus. But recently it's been shown that some of these cells can actually be generated in the periphery. So this is not really clear, but they think most of these cells, again, are generated in the thymus. Some can be generated in the periphery. Important point here is that T regulatory cells don't really affect T cell activation. They seem to affect the sustained response. So once you get the response, once the response is started, you have the activation of these cells, then the T regulatory cells can come in. And it's thought that the major role they play is preventing chronic and potentially damaging responses. So again, if you have activated cells hanging around for too long, and this can lead to various autoimmune disorders, as well as uncontrolled inflammatory responses. So shown here is a picture of the development. Typically, you have a T cell precursor. We discussed a lot about this development, the conventional T cell, which can then lead to naive T cell, which plays a role in development of a number of different T helper cells, for example. In addition, there is a second population, or the T regulatory cells, that can develop in the thymus. Important characteristics here is that from the beginning they express CD25, CD4. They also express FOXP3. Now, FOXP3, it's not really clear what the role of FOXP3 is in the role of the regulatory cells. Some studies show that FOXP3 can downregulate transcriptions of certain genes, but it's not really clear what the role of this particular protein is. However, they know that if a cell is expressing FOXP3, more than likely it's a T regulatory cell. The conventional T cell, once it gets into the periphery, some way this is not really clear, but it can actually develop into a T regulatory cell. So the main ways is development of the thymus. However, some can develop from naive T cells in the periphery. T regulatory cells in the periphery are anergic, or in vitro are anergic. That basically means that they don't respond to activation stimuli. It's very hard to get these cells to proliferate. They don't have the unique characteristics of a Th1 or Th2. So if you look at cytokines produced, their patterns don't resemble Th1 or Th2. They're very unique from these two cell populations. Again, as we discussed, they express FOXP3. To my knowledge, the exact function of this FOXP3 has not been elucidated. They just know that T regulatory cells express it. They also know if you get rid of it, so if you take a mouse, delete FOXP3, these mice will develop a lot of autoimmune disorders or uncontrolled immune responses. So FOXP3 expressed by T regulatory cells plays some important role in their function. The exact mechanism by which T regulatory cells are able to downregulate the immune response is not really clear. There are some possible mechanisms that have been proposed. They may act in a cell contact dependent manner by competing for stimulatory ligands. It's thought they may actually compete for essential growth factors. So they may actually gobble up important cytokines such as IL-2. By doing this, IL-2 will not be available for the naive or other cells of the immune system. It's thought that they actually may come into contact with the cells and actually transmit some regulatory signal, and as yet uncharacterized. So again, it's just a hypothesis. Some people suggest that cell contact may not be critical, that maybe T regulatory cells actually produce suppressive cytokines. Two such are IL-10 and TGF-beta. Some evidence in vivo, but in vitro, they can't show this. Another possibility that these T regulatory cells may act somehow to actually affect the APC. They may negatively affect the APCs. And there's two potential hypotheses here are they may act by generation of immunosuppressive metabolites. So maybe T regulatory cells produce something that then interacts with APCs and prevents them from being able to effectively present the antigen. Similar here, they may produce factors that, again, affect the ability of the APC to present cells efficiently, present antigen efficiently. Yes?
Do they think that they're active all the time, or do they know what they...
That's a really good question, but I don't know that the answer is completely clear. Some people, if you look in the recent literature, some people think it's antigen dependent or it may require some type of stimulation in the immune response. And some other people think that it's just they're there all the time and they act to downregulate the immune response. Again, I got how to put this as a disclaimer. That some of these may happen and some of these may not, or there may be a combination of these. So you may have production of these immunosuppressive factors, and you may have other ways of suppressing the response. So again, they may act in one of these ways, they may act in all of them. It's not really clear yet. So shown here again is the cartoon. They were T regulatory cell. It's thought that they can affect APCs. Again, they think that by affecting the APCs, they can cause the APCs to produce some suppressive factors. I think also that they may affect APCs in their ability to present antigen to the other T cells. And it's also thought that they may actually be able to directly affect the T cells. So they may either produce factors that directly downregulate the T cell activity, or they may act by just gobbling up factors that are necessary for the T cell activity. Important point here is that I'd like you to know is the phenotypic characterization of CD25, CD4, CD25, and they act to downregulate the immune response. Okay? I don't think it's important to memorize all the mechanisms because at this point, this mechanisms have not been really clarified. So, okay, that's it. Have any questions? Again, that's a lot of material for one lecture. Please feel free to email me